Mhm. All right. Well, I'd like to thank the organizers of the Myelin Neurotech Research Group for having me. I'm excited today to talk about how our unique software is revolutionizing shape measurements for discovery and diagnostics, right, in biomedical science. And I'll talk today in particular more neuroscience and neurobiology applications, since this is a neurotech group. So my name is David Nguyen. I'm CEO of Brainscanology. And I invented an algorithm that measures subdimensions of spatial information that the eye cannot see and that previous metrics cannot measure. And I turned that algorithm into a software called ShapeGenie. So this talk will have 4 parts. First, I will talk about why we study shapes in nature. Then I'll talk about examples of capturing spatial context for understanding Alzheimer's disease. And then I'll talk about measuring shapes of brain organoids in culture. And lastly, I'll talk about how we reinvented the clock draw test using our algorithm, right, to develop a at-home screening tool that detects early signs of dementia. So first, why do we study shapes in nature? Well, in biochemistry, there's a phrase that goes, or a mantra that goes, structure is function. That is, if you find recurring patterns, right, of structure, then you're likely to find distinct functions, right, with each pattern. And this mantra turns out to be true not just in biochemistry, but throughout all scales of biology. And here's an example of how our traditional metrics, such as area, volume, perimeter, etc., right, is useful, but also highly uninformative in very important cases. So in front of you is 2 coffee mugs that are identical, right? They both hold the same volume of hot water, right? But volume cannot tell the direction of gravity, right? The spatial context in this case is gravity, right? And so area and volume, right, though they're great and useful in biology, right, they're agnostic to spatial relationships, because in one case, one hand is about to be scalded with hot water, right? In another case, the hand is safe. But if you ask volume, tell me which condition is safe and which one is unsafe, volume says there's no difference. You see that? So volumes, as useful as it is, cannot capture basic spatial context as relevant to health and safety. It turns out spatial context is important at all scales within nature, right? In panel A, even amino acids, right, on Earth, at least, they have to be left-handed in order to be integrated into proteins. In panel B, you see that even at the tissue level, glands follow a certain recurring pattern of structure. When they invade into each other, we call that cancer, right? And even at the ecology levels, right, there are certain trees that there's a phenomenon known as crown shyness in panel C, such that trees, they will not touch each other, right? So you cannot fully understand the growth pattern of one tree unless you understand where its neighbors are. And then lastly, in D, right, another example of ecology, spatial context and ecology, right, moss and lichen prefer the shaded side. So they're avoiding the most direct sunlight, right? And if we want to understand growth patterns of these organisms in their natural setting, we have to understand the spatial context that they're trying to avoid direct sunlight. So this goes, we then move on to talk about how ShapeGenie, right, measures shapes in ways that are very unique. Well, the overall idea is that here we have 2 shapes, on top a crescent and the bottom just, you know, a closed blob shape. We have 2 choices of grid systems. At the top, we overlay a parallel grid of lines. At the bottom, we overlay a radial grid of lines. And what we do is, excuse me, we find all the points of intersection between our shape and our grid lines. And that allows us to measure distance. That is the distance of each spot. At the top, right, each spot to the vertical dotted line, which is our reference line. At the bottom, the reference point is the red dot, which is the center of the bounding box, the smallest box that encloses your shape. And so having 2 points, we can measure distance between every single intersection point. What we do next is we turn the intersections we have in step 3 into a linearized form in which the x-axis is just the grid line position, and then the y-axis is just a distance to your reference point. Okay, and if any line that touches 2 points, we sum those 2 distances together to get one value. Thus, for each value on the x-axis in panel step 4, there's only one y value. And once we get data in this form, which is called a discrete sinusoid wave, we apply what's called a fast Fourier transform and look at a different dimension, right? We look at the frequency dimension. And so what we have now in step 5 is a multidimensional representation of the shape we began with in step 1, right? And a multidimensional representation, the difference between that and a scalar value, a scalar representation, which is just one number, is like my age in years. That's a scalar, right? But my birth date is a multidimensional representation of my age, because you can calculate my age from my birth date, but you also know my month, my date, and the actual year, right? So multidimensional data is more complicated, but it brings so much more information than just scalar data, right? But to simplify the data further, right, as scientists, we like to create indices. So to turn step 5 into one simple number, there's many ways of doing that. One simple way is to sum all of the bars together to get one value, right? And we call that the sum all bins index. And that's one way of representing the shape that you began with in step 1 with just one number. So there's many ways of calculating indices, and we calculate it depending on the shapes that we are measuring. Okay, but that's how we simplify the shape even further, by calculating a scalar value through an index. All right, and so what's also special about Shape Genie is that it allows us to measure things that typically people throw up their hands up and give up against, right? It's complexity, or what people call randomness. Right here, as an example, is just micrographs of some fiber structure, and the fiber arrangements are very haphazard, right? But there's a difference between disorder and randomness. It's really hard to measure randomness objectively, but if you call it disorder, then you can also derive a shape index. In this example, in the middle panel, I just outline only one side of each fiber, and when I extracted the mask of my outlines, as you see in the far right column, if we apply the parallel grid system to this, we can generate precise numbers representing the density, okay, of one image compared to the density of another image, right? And I point this out because most of the images in biology is very complicated, right? And it's without now we have the tools to measure that complexity in very precise ways. So we no longer have to throw our hands up and say, oh, it's just heterogeneity, we can't measure it. Now we can, right? But it's not an intuition that comes naturally to people. Thus, I pointed out. Okay, and next, a very special thing that Shape Genie can do is we measure what's called pure shape. So on the left, I have two objects. They're the same. I duplicated them. I just enlarged the blue to become the red, right? So on the left, it's the same shape, but different sizes, okay? But what we can do with Shape Genie is we can measure the pure shape. That is, I find an objective way to resize them. In this case, I found the longest internal length of each object, and I resized that length to be the same. Okay, so you see on the right side, the objects are both now the same shape and the same size, right? So, but when we resize it, we always constrain the aspect ratio, so a square doesn't become a rectangle, so to speak. And when we do that, what we do is we measure shape apart from the effect of size. And this is something impossible until now. And in many instances, as you will see, right, we're finding really amazing things and much cleaner data than traditional metrics such as volume, area, perimeter, etc. Okay, so Shape Genie, right, is we have a freemium model that is anybody can create a free account. We did that so that students, right, even high school students can create their free account and learn how to use Shape Genie, right, for their science fair projects, research projects, and of course for research labs who want to explore how Shape Genie can benefit them. All right, so the link is on the screen. And we have, if you visit our website, excuse me, we have a whole page dedicated to how to use Shape Genie, right? In 6, 10-minute videos about. We even give sample practice files. You'll learn how to use Shape Genie. And on the right side, we have another page dedicated to our many use cases. Today, I'll talk about 3, but we have many, many more, which we could continually add to. So you can see, examples how we apply Shape Genie to questions that are most similar to your research question. So the first example we'll talk about of Shape Genie in health sciences, neuroscience is capturing anatomical spatial context in Alzheimer's disease. So here is a cross-section of a structural MRI of the brain. And we measured these the lateral ventricles here outlined in red and blue. Okay, and we found that when we measure the shape as we extracted them, you see on the right, we applied the parallel grid system to it. And when we compared normal patients, cognitively normal patients to Alzheimer's patients, we found a large difference, right? But when we compare this measure from in MCI patients, that is patients who have mild cognitive impairment compared to Alzheimer's, the difference was very weak. And I found this to be highly, unsatisfying, because visually I could pick out a difference between patients who had mild cognitive impairment versus Alzheimer's. And yet quantitatively, right, it was a very weak difference. And so I went in and I explored it more, and I realized, what if, right, we added an anatomical marker? So here I added a green line here of the same length, right, and starting at the same position in every patient. And what this line does is it captures the spatial context of the center line of the brain. And it turns out when you do that, right, we then found, right, a big difference between patients who have mild cognitive impairment versus those who had Alzheimer's, right? And the only difference we did was what? We added an anatomical marker. You see that? To capture the spatial context. Turns out these caverns, when they are enlarging due to Alzheimer's, they enlarge away from the center, right, of the brain, right? They're not just enlarging in place, but they enlarge away from the center. So this is an example of how Shape Genie allowed us to capture spatial context, right? Because again, with the hot water analogy earlier, volume does not capture spatial context. It doesn't know where gravity is, and it doesn't know where the center of your brain is. And for humans who have bilateral symmetry, right, spatial context is actually quite important for a number of diseases. Okay, and so here it's just to point out that, yes, we added an anatomical marker, right, to capture the midline, but we have to add the same amount of information to every single sample to be consistent across all samples. And here's an example of what we found here. In this case, we measured the shape of the lateral ventricles in this particular slice of the brain. As you can see in blue, this is cognitively normal patients, and then in orange is people with Alzheimer's. As you can see that we found differences in every half decade, okay, of life in our study group, or this is archived data. And as you can see here that for us, we're building a machine learning model based on this data to classify Alzheimer's versus cognitively normal, right? And for us, we have to limit the age bin of our models to be only 5 years, right? And it's the opposite problem of all the other machine learning papers published out there doing the same thing on structural MRI, right? They need large sample sizes to have accurate models, and therefore their age bins range from 10 to 20 some years, right, per model. But for us, we have the opposite problem, right? What we measure is so precise that we realize that if we don't narrow our age bins to just 5 years, half decades, our models become inaccurate. So this goes to tell you that what Shape Genie is measuring, right, is something that even the best deep learning models aren't picking up, right? Because the current deep learning models in the literature, they require a very large sample size and thus very large age bins. So next, I'm going to talk about an example of shape analysis in neurobiology, that is brains on a chip or brains in a dish, that is 3D organoids. And so we analyzed this image from this particular preprint, as we found it to be interesting. And here, if you examine the shape of these organoids, so this lab, what they did was they created mutants, variations of an ATM knockout, and they grew brain organoids based on these cell lines. And as you can see that going from wild type, right, to the right, the knockout that has the most severe phenotype, that the shape of the edge of the organoids become more and more jagged, right, as you go from wild type, right, to the genotype on the left. And in their paper, they measured area, right? And as you can see here, I've taken it from their preprint, that they found no difference in area, right? Even though visually to the naked eye, you can see that, oh yeah, I mean, there's definitely a difference in the jaggedness of the shape of these organoids, right? Which is important because brains have folds, and of course, brain organoids would naturally want to form folds, right? Because that's what they're supposed to do as the brain develops. And yet area is unable to capture this. And so what we did is we went in and we outlined here, we segmented the shapes of these organoids. And what we did was we always standardize orientation before we do our shape analysis. And the way we did that is we found the longest internal line, and then we rotate the object, the organoid, so that it's vertical. And then we found the smallest bounding box. And all of this can be done in Shape Genie. And we do this because nature is naturally polar, right? There's North Pole, South Pole, anterior, posterior. So when we align shapes according to these natural poles, we find that we get cleaner data, right? And thus we have the step of normalizing the orientation. And as a result, at the top, when we measured these shapes at scale, that is, we measured them in their original scale in the images relative to each other, we only found 2 pairs that had statistical significant differences. But when we measured the pure shape, as you see in the bottom row, right, we have 3 different indices we calculated, and suddenly you see more statistical significance, right, between these data. And this is with a very small sample size, right? This is, all right, we just analyzed this image. You see that? They had the advantage of triplicates, right, and replicates. We didn't, because we just, analyzed this figure as a mini pilot study to prove a point, right? And even with a small sample size, we see statistically significant differences, right, in measuring pure shape, right? Something that only Shape Genie can measure, right? So clearly there's a difference between shape and size, right? And it's something that the eye can pick out intuitively, but traditional metrics just are unable to detect. Okay, now moving on to part 4, the last part of my presentation, right? So what we did was we applied Shape Genie, our shape analysis tool, to something called the clock draw test. Okay, and we created an app that allows people to use our clock draw test once a month to detect early signs of dementia. So the clock draw test is a classic, diagnostic tool in the tool belt of psychiatrists and neuropsychologists. So how a person with dementia is unable to draw a normal looking clock, as you see examples on the screen. And drawing a clock with 12 numbers and 2 hands turns out requires a lot of planning and reasoning, right? It tests multiple things at once. And thus this has become a very useful tool in detecting cognitive impairment. But the reason why it hasn't been deployed more widely is one is that you need a live person to watch you draw it, okay? And 2, it's really hard to interpret the clock draw unless the clock is obviously weird, right? Because, some people are just bad at drawing, or they had wrist injuries. And so therefore, their clock isn't as perfect a circle as, should be, but that's not dementia, right? And so, so the subjectivity with which, clock drawings are scored is also a limitation as to why we haven't, we don't use this as a screening tool that's purely self-administered. Well, Brainscanology solved that problem, right, with Shape Genie, because we can measure pure shape. So no matter when somebody draws clocks, okay, on a piece of paper, right, multiple times, right, say once a month, no matter how they draw it, large or small, right, we can resize it and measure its pure shape, right? And so this makes, this allows us to objectively measure the normal variation in how people draw clocks, right? Because unless you're an artist, your clock's going to look slightly different every time you draw it, and that's normal. And so this allows us to, okay, develop this app to detect early signs of cognitive impairment through clock shapes. And here is a hypothetical series, right, of somebody quickly progressing within a matter of months from cognitively normal to dementia. As you can see here, the shape begins to elongate, and then eventually the circle doesn't even close, right? And the hands no longer connect, and the distribution of number becomes very different, okay? So the purpose of this test is to be an at-home screening tool, such that once used over time, you will begin to see how your scores are moving into the abnormal zone, such that it is a warning for you to seek professional help. And so here, each time somebody draws a picture on a piece of paper, takes a picture of it, and then uploads it to their app, the phone, right? We will send them an objective score report with 7 numbers, and each number has a range of what we define as the normal range and the abnormal range. Okay, thus we've turned the scoring of the clock draw test into something that is objective. And we allow, of course, monthly tracking and analytics so that you can request, if you have 12 scores or 12 submissions, you can request that we send you analytics, and we will graph out those 7 scores so you can see the trends, which ones are moving in the abnormal direction or which one is staying in the normal direction. Okay, and so that is the end of my presentation on the usefulness of Shape Genie or shape analysis in neuroscience, both in basic science and clinical science. If you have any questions, feel free to reach out to me at dave@brainscanology.com. And now I'd be happy to answer any questions.
Cool. I apologize for the background noise again, but yeah, I have a few. I'll just ask one and then see if Gabe has any. But has the Alzheimer's detection app, has that been used to detect any Alzheimer's yet? Has anybody been able to use it for themselves successfully?
Yeah, we've had several hundred downloads so far. We released this recently, and 2 of them, at least the early ones, the first submissions, we found oddities, right? And one of them, that person actually reached out to us and thanked us for confirming their suspicions that they had a family member who was really beginning to stop sentences midway and be very forgetful. And that person did 2 clock draw tests and 2 submissions, and it would confirm to the family that there was cognitive impairment going on. I'm saying so. In that, yes. In terms of us, we've yet to detect somebody who is gradually transitioning, right, to early stage, right. We're still waiting for that to happen as people continue to use it, and it is meant to be a screening tool as opposed to a one-time diagnostic. And there are good reasons for that, because a lot of non-dementia related reasons can cause you to draw a weird clock one time. For example, UTIs, urinary tract infections, are very common in nursing facilities, right? And UTI causes delirium, right? But delirium is momentary, right? Once you heal from the infection, right, you're no longer delirious, and so your clock score should revert, back to normal. You know what I'm saying, right? Or even dehydration or low blood sugar from diabetics, right? That will cause delirium too, right? And thus, as a standalone, purely self-administered tool, right, it cannot diagnose dementia. You know what I'm saying? But used as a screening tool over time, it'll give you indication whether or not you have persistent cognitive problems, right? Which is a warning signal for you to seek a professional diagnosis.
Okay, that makes sense. Thanks.
Are there any open data sets that you're able to kind of like retroactively see the sensitivity and specificity of this test?
Yes, we are seeking to get access to it. We're currently applying to, there's certain data sets where labs have uploaded, hundreds of clocks from, various people. So we're in the process of doing that right now. Our own, we're doing it in-house, right? Our own, from our own users. And thus far, we've only had cognitively normal users, right? Other than the ones who've, pointed out that, they suspect their family has, cognitive problems. But of course, with those who are cognitively normal, it's 100% accuracy, right? I'm saying so. But we're currently tracking that with our users, and we are in the process of preparing to write a grant to do a formal study in a medical setting in which we actually apply this to people who are actually transitioning from normal to MCI or from MCI to Alzheimer's. You know what I'm saying? So I think so that is in the works.
Awesome. And I have kind of a question that dovetails with that. Does this have any place to integrate with other machine learning techniques? The numbers and that Shape Genie is describing these shapes or phenomena with. Does this go into like a linear classifier, or does it go into other models?
The answer is yes, it can. The output of Shape Genie is just a bunch of numbers, right? And if anything's in the form of a number, then it's applicable to data science techniques for you to integrate with any other data. In fact, a number of our partners or pending partners are interested in integrating Shape Genie into their pipeline to enhance their models or to build their models, right? And so.
Oh, sweet. I have some other let's see. There's a couple other technical questions. I'm wondering how much. I'm sure I see a lot of the kind of intrinsic differentiating things to like other image-based classifiers and metrics you could drive using like a ResNet. What do you think the primary distinguishing factor though in between that and other computer vision techniques?
Yeah, the primary difference is that our the LCPC transform, right, in Shape Genie, what it does is it captures spatial context in a way that other methods can't. And the advantage isn't in the actual data science models, right? The advantage we provide is how we extract the data before you even apply the model to it, right? For example, the example of the hot water, right, or hot coffee mugs. The volume, right, is just a number that you extract from the volume of liquid in the cups. Okay, but volume can't tell the difference between a cup that's right side up versus upside down. Volume doesn't know where the direction of gravity is, right? So when we give you a number that captures the direction of gravity, right, we give you better data, right? So that even, pick your favorite model, right? And that model will likely be more accurate because we give you cleaner data to begin with. That makes sense. So that's the main advantage, actually. It's not the LCPC transform is a data extraction algorithm. It's not a classification. It's not a machine learning algorithm. It produces data so that you can then build a machine learning algorithm. But and so yeah, so really you can pick your own your favorite machine learning classification model, right? And as long as you have good data, that model is going to work.
I see. Sweet.
I was wondering, did Brainscanology invent a lot of these shape analysis tools, or were they popular in any way beforehand? Like, did Brainscanology or is Brainscanology is like an implementation of them, like a way to make them an accessible tool for researchers?
Yeah, and so I invented the LCPC transform, and we turned it into Shape Genie, which is like a user-friendly, no code needed software that anybody can use, right? And so yeah, so I mean we are the pioneers of it. I guess I'm the pioneer of it, and it was something completely new actually, and it came because of my background in pathology, cancer biology, pathology, and digital pathology. So actually, my 3 years ago or so now, my favorite high school biology teacher who inspired me to go into science, I found out he died of an aggressive colon cancer that came back after 3 surgeries, and I was unable to visit or attend his memorial service in Los Angeles because I don't live there anymore. So I wanted to commemorate him in some way, the impact he had on me and many of students over his 30-year career. And so I spent a lot of time, several days, looking at colon polyp images, histology, and figuring out how can we measure these colon polyp shapes more precisely, so we can grade them more precisely beyond just 1, 2, and 3. And so it was based on those images that I derived, what is now the LCPC transform, right, to commemorate him. And informally, his name is Dwayne Nichols. And my last name I'm Dave Nguyen, and so I named it informally the Nguyen-Nichols transform, right? But the formal name is the LCPC, which is linearized compressed polar coordinates transform. But that's how it came to be, right? To solve that problem in digital pathology. And it turns out it works on every single shape out there beyond just polyps, right? And tumor nuclei. You know what I'm saying? And so that's what we realized, that we should turn it into a software that could be commercialized and user-friendly.
Cool. So there are a lot of different ways you could transform shapes to generate data on them. Why is this method in particular especially useful?
One is it captures spatial context in ways that other ones cannot. And two, it's just novel in terms of measuring shapes intuitively, right? Because there's many ways of transforming data, right, to calculate indices. You know what I'm saying? But this method turns out to give good coverage and versatile coverage, depending on what shape you're measuring. For example, here, the two grids that we allow users to pick is the parallel grid system at the top and the radial grid system at the bottom. There are other grid systems you can use, but to simplify it, we offer these two. And the advantage of a grid system is spatial context, right? For example, how we measured the center line of the brain, right, when measuring the shape of these caverns, right? Something that, surface area, volume, signal intensity, right? None of those metrics knows where the middle of the brain is, right? It just measures, the shape you're interested in space.
Okay, yeah, that makes sense. And then I guess I think maybe the last question I have. Sorry again for the background noise. There's a. Do you have any way to guard against manipulating the way that you analyze your data to fit your expectations? Because that's another common cause in or another common issue in machine learning models is you make the model in a way that you think it's working because it agrees with what you think should be right. How do you prevent against that in this context where you analyze the data so that it generates a value that looks right?
Yeah, and the answer to that is just like anything else, external validation, right? Models are easily overfitted, especially deep learning models, right? And even in our case, even though what we measure is very interpretable, right? We know exactly what we're measuring and why. You know what I'm saying? We still have, once you build a model, you have to validate it on external data sets, right? And ultimately, that's the way to do it. And that's how you guarantee against overfitting. Mm-hmm.
Okay, cool. Thanks. Gabe, did you have anything else?
No, I think I'm all set.
All right, cool. Thank you so much. We really appreciate this, and we'll definitely share it with all our members, put it up on our Google Drive and YouTube channel.
All right. Thank you. I look forward to sharing it too. All right.
Yeah, thank you so
have
much.
a wonderful day.
You too.
Thank you.
Thanks.
Take care.
Bye.